Since the human genome was sequenced, scientists have had the instructions for the structure and function of every component of every cell in the body at their disposal. Occasionally, within these instructions, errors or changes to gene sequences can arise which may cause disease. But investigators have lacked a practical and precise way to edit gene sequences to treat and potentially cure disease. That is, until now. CRISPR is a precise genome editing tool which enables scientists to work inside cells and make specific changes to genes. It is a two-part system comprising a single guide RNA which directs the Cas9 nuclease to cause a double-stranded break in the matching DNA sequence. When Cas9 cuts the DNA, it triggers the cell's natural repair enzymes to fix these breaks. It's during this process that the target gene can be modified by adding to, disrupting, or changing its genetic information. Or instead of rewriting the gene sequence itself, different types of CRISPR can be used upstream to switch genes on or off. By simply altering the composition of the guide RNA, different precise locations within the genome can be targeted. AstraZeneca is employing CRISPR within its drug discovery platform to help speed and refine our understanding of disease. By changing the activity of a gene in a biological pathway and examining the impact of this change, we will discover novel therapeutic targets. Precise control over the genome enables us to accurately model human disease in cell lines in weeks rather than months, and these models are used to screen compounds to find new, safer medicines. Ultimately, CRISPR will help us to reach parts of the genome that traditional medicines cannot, and may enable us to alter the genetic code to correct genetic errors and cure disease. Together with our in-house CRISPR team and with our world-renowned academic partners, AstraZeneca is pioneering technology to genetically edit the way to better treatments for patients.
Genetics, what can it mean? The ability to perfect the physical and mental characteristics of every unborn child.
Of genetic engineering to choose the gene.
In the not-too-distant future, our DNA will determine everything about us. A minute drop of blood, saliva, or a single hair determines where you can work, who you should marry, what you're capable of achieving. In a society where success is determined by science, divided by the standards of perfection, one man's only chance
How do you expect to pull this off? I don't know exactly.
is to hide his own identity
This is the last day that you're gonna be you and I'm gonna be me.
by borrowing someone else's.
Congratulations. What about the interview? That was it.
Do you think
Geneticist Craig Venter spent 15 years and $40 million to create the world's first synthetic cell. Using chemicals to assemble 1.1 million letters of genetic code, the team built a bacterial genome from scratch, and Venter has speculated that bacteria could be created which absorb carbon dioxide, produce biofuel, or even vaccines. In 2014, biotech company Synthorax went one step further by expanding the genetic code of life. Instead of the 4 chemical bases of DNA that make up all known living organisms, Synthorax added 2 more. The company has reportedly already created a new form of E. coli never seen before and hopes their synthetic life forms will improve medicine. 5. Super photosynthesis. More than 40% of the world relies on rice and wheat for food, but by the time the human population reaches 10 billion in 2050, the Earth will be 394 million tons of rice short to feed everyone. However, the C4 photosynthesis process captures CO2 and concentrates it in special cells inside plants' leaves. Early tests have shown this can increase the yields of these crucial crops by 50%. According to MIT, this genetic advancement should be available in the next 10 to 15 years, in time to save millions of lives. 4. Gene editing. In 2015, Chinese scientists genetically modified human embryos for the first time in history. Researchers used 86 embryos, successfully editing out a gene which causes a fatal blood disorder in 28 of them. However, they also caused unintended mutations. Altering embryos is largely illegal and highly controversial, but gene editing could help to prevent more than 10,000 diseases caused by flaws in DNA, potentially saving millions of lives. It could be used on adults as well, for example by making them immune to HIV by modifying their blood cells to give them natural protection.